Hello, everyone. This is Mr. Friedoff. I'm going to be talking about Chapter 8. This is going to be our first chapter on something called cell division. Up until this point, we've been talking about cells a little bit. We've been talking about the processes that go on within cells, like photosynthesis and cellular respiration, and how energy is transferred between those different processes. During this chapter, we're going to kind of switch our kind of focus on the content to how then cells divide. So we're still talking about cells, but we're going to first talk about how mitosis creates a specific type of cell division. So with that being said, I'm going to first talk about something called, or why do we need cells to divide? So I always like to pose the question of what is cell division and why do cells actually need to divide? If you think about it, you're made of trillions of cells. Why aren't you just made of one really big cell? Why do we have to have all these small cells that just continuously divide? So we're going to talk about that in this chapter. But cell division overall is just the division of cells from one cell becoming two cells. If you look here, we can see one cell here. It's going to split off and become two cells. So that is what we're going to be focusing on in this section. Now, one of the reasons why your cells need to divide is to replace damaged or old cells. These cells may, have been damaged through whatever happens, but they're just damaged and they need to be replaced. Growth of an organism. So organisms obviously need to grow. The animation you see here is a frog embryo growing and developing. You can see it's actually just splitting up right now, but it's just the growth of an organism. And also for reproduction. We're going to talk about how cells divide through reproduction in the next chapter a little bit more, called meiosis. But we're going to be focusing on these two first reasons for mitosis. We're also going to talk about in section 8.1 something called surface area and volume. Remember that surface area is the outside of a cell, while volume is the inside of a cell, or whatever we're looking at. So if we're looking at a box, the outside of it is the surface area. The inside is the volume. You can see that here in this image. You can see the surface area being the outside, the volume being the inside. What happens when cells divide is they increase their surface area to volume ratio. If a cell just keeps on getting bigger and bigger and bigger, that surface area to volume ratio goes down. And the reason why we want cells to divide and remain really small is it's more efficient to move things into and out of the cell when they're smaller. If you look here, it's really hard to get things into the nucleus going back and forth from this really big cell. But it's really easy to get things into and out of the nucleus in this smaller cell. It's because the smaller cells have a higher surface area to volume ratio that we can get things into and out of the cell a lot easier. So the reason why cells divide is it increases their surface area to volume ratio. We replace old and damaged cells, and we also help with the growth of organisms. So those are the three main reasons why cells divide. Hello, everyone. This is going to be the second section in Chapter 8 where we talk about mitosis. And in this section, we're going to be talking about chromosomes. Now, just a little bit of a preface before we get into what chromosomes are. Remember that DNA is the instructions for every characteristic of an organism, and DNA is going to be located in the nucleus. I feel that by 9th or 10th grade biology class, we should know a little bit about DNA, so I'm not going to spend much time on here. But remember, DNA is made of this double helix that we should be able to recognize. Now, this double helix is going to coil around these things called histones, and these histones are going to make these kind of fibers. They're going to make this thing called a chromosome. And these chromosomes are, again, just condensed and organized structures of DNA. Here's another way of looking at a chromosome. You can see this is all those fibers of DNA. And we have chromosomes that have 2 chromatids, and we have chromosomes that have 1 chromatid. So you can see here, this is a chromosome with 1 chromatid. This is 1 arm. This chromosome has 2 chromatids, so it has 2 arms. In the middle, where the arms meet, is something called the centromere. This is a region where the chromatids are going to attach. One single chromatid still has a centromere. It still has a middle region here, but this is what a centromere is. It's just the center. So think centromere, center. When chromatids are identical and they are attached here at the centromere, we call them sister chromatids. So they are attached here at the centromere. They're going to be identical. And this only occurs after something called replication, which we're going to get into later on. But they're attached at the centromere. You can see here the genes here are identical between these sister chromatids because they are attached at the centromere. Lastly, we're going to talk about something called homologous chromosomes. Homologous chromosomes are when there are pairs of chromosomes with identical features. Now, these will not be sister chromatids. It sounds like they should be because they look identical, but this would be A homologous chromosome, or I'm sorry, a set of homologous chromosomes, because you get one of these chromosomes from dad and one from mom. You would typically have the same genes on each chromosome, but you might have different versions of the genes. You might get like a brown eye gene from dad and a blue eye gene from mom. So they're the same gene, but just different versions or different like flavors of the same gene. Typically, humans have 46 chromosomes and 23 pairs of these chromosomes. You can see here what those homologous chromosomes look like. You can see they're homologous because they have the same genes in the same area, but you might see here, hey, these are different colors representing different versions of the gene. Now, what we do with those chromosomes is we can say, hey, there are 23 pairs of these chromosomes which make up a human karyotype. You can see these chromosomes align because they're the same length and they have the same genes in the same order. All right, so that's the end of section. All right, section 3 is going to be about the cell cycle. And in the cell cycle, we have two main parts. We have interphase and M phase. The diagram here on the right here is going to be an image of the cell cycle. You can see it looks like a circle because cells will just continuously go through this cycle. So once it goes through the interphase, it's going to enter M phase. Then it's going to re-enter interphase. Then it's going to re-enter M phase. So it just keeps on going around and around and around. But during interphase, normal cellular functions occur. This is just like most of your cells right now are in interphase. They're just normally going through their daily, activities. They're just normally doing what cells do. During M phase, though, the cells are going to divide. You can see M phase here. This is the part of the pie or the part of the pizza where the cells are going to be dividing, and these are going to be called somatic cells or body cells. Interphase is made of three parts. We can see G1, S, and G2. During G1 phase, the cell is just going to grow and duplicate its organelles because it just got done dividing. During S phase, replication is going to occur. This is when the DNA is going to be duplicated. It's going to double. And during G2 phase, this is going to be where we prep for mitosis. The cell is going to continue to grow, and we're just going to get ready for that M phase. Let's show you what that replication looks like. You can see the DNA here, and what's going to happen is the DNA is going to double. Oh, by the way, the DNA here is going to be unbound or it's not going to be condensed. It's going to be like spaghetti. But you can see here, replication is where the DNA is going to be copied. It's going to double in amount. You can see here, this DNA, we have one chromatid per chromosome. Each chromosome is going to have two chromatids afterwards. You can see what that looks like here, where the DNA is just doubling itself. It's replicating. It's duplicating itself. So that's what happens during S phase. During M phase, this is again after interphase. This is where mitosis begins. This is where the division of the nucleus happens. And this is also where cytokinesis, which is the division of the cytoplasm, occurs. So going back here a little bit, let's just review. During interphase, the cell is going to grow and go through replication. During M phase, the cell is going to divide. And when that happens, you're going to get two new cells. Hello, everyone. We're going to talk about section 8.4 here. We're going to talk about M phase. We just got talking about the interphase part of the cell cycle. This is, again, G1, S, and G2. This is where, again, the cell is just going to go through normal functions. But remember that the cell here is going to start dividing in M phase. So M phase stands for mitosis. And we're going to have two parts of M phase. We're going to have mitosis, and then we're going to have something called cytokinesis here. But again, this occurs after the cell goes through interphase. Whenever the cell gets done with M phase, it's going to re-enter interphase, and then it's going to go through G1, S, and G2, and then it's going to go back into M phase, and then it's going to go back into interphase. So these are just going to cycle back and forth as the cell continuously divides. At each one of these phases, I'm going to show you a diagram that looks like this. This diagram is going to help us see what is actually occurring in the cell. So you can see here, this is a cell. You can see these are our chromosomes, and they are going to start to condense. I have one word here underlined to help you remember, hey, if you remember one thing about this phase, this is what I want you to remember. Chromosomes condense. The spindle fibers are also going to form. These are going to be what move the chromosomes around, and the nuclear membrane or the nucleus starts to break down. So you can see what that nucleus looks like here. It's going to start to break down here. The second phase of mitosis or M phase is going to be called metaphase. This is where the spindle fibers are going to attach to the centromeres of the chromosomes. Remember, that's the middle here. And they're going to move them in the middle. So again, these spindle fibers are going to attach to the chromosomes, and they're going to move them in the middle. I have a line here. I also could sometimes underline middle. So the chromosomes are going to be lined in the middle here. So in metaphase, think middle. You can see these chromosomes are aligned in the middle. The spindle fibers are going to be attached here at the centromeres, and they are getting ready to divide. Anaphase. When I think of anaphase, I think of separating. These chromosomes are going to be split by the spindle fibers shortening. So you can see these spindle fibers are going to shorten. They're actually going to tear these chromosomes apart. Now, you might think, well, isn't that going to split the DNA? Well, yeah, it will, but we already doubled the DNA in our last step in S phase of interphase. So what happens here is these chromosomes are going to be split, but they're still going to have the same amount of genetic information the cell started with. So in each cell here, we're going to have 4 chromosomes. You can see 4 chromosomes here and 4 chromosomes here. So each cell is going to have the complete set of genetic material it needs to survive. Telophase. This is when the nuclear membrane or nucleus starts to reform. Chromosomes are going to unwind, and this is where we start to see 2 new cells. You can see how this chromosome, or the cell here, is going to start to be divided here in the middle. The last phase of M phase is called cytokinesis, and it's really important to remember that cytokinesis is not a part of mitosis. Mitosis is only the division of the nucleus, while cytokinesis is a division of the cytoplasm. So cyto, think cytoplasm, think mitosis, the nucleus. So you can see at this point the M phase is going to be complete, but mitosis is also going to be complete. And we get 2 new cells with the same exact genetic information. All right, now we're going to talk about just the products of cell division and the products of mitosis. So in the last section, we talked about how mitosis occurred. And again, chromosomes are going to condense, they're going to be split, and then the whole cell splits. That is mitosis. So what happens at the end of mitosis? Like, what do we actually produce? Well, we're going to produce 2 genetically identical daughter cells. Now, when we say daughter cells, we're not saying they're female cells. We're just saying they are like the offspring of the parent cell. This animation does a pretty good job of showing what that looks like. You can see again here, we're going to go through all the phases of mitosis. It's going to restart here. Prophase, the chromosomes condense. The chromosomes are going to lie in the middle. They're going to be split apart. And they're going to reform the nucleus. And then cytokinesis is when the cell starts to split. Each of these cells will be genetically identical, which should make sense because we're making body cells, or they're also called somatic cells. We want all of your somatic or body cells to be the same. We don't want different cells. So we want all the same genetic material to be there at the end of mitosis. Now, it's important to remember some things about these cells. These cells are going to have the complete set of chromosomes. We call those cells diploid or 2N. These are cells that have at least 2 sets of chromosomes per cell. These are your body cells. Haploid cells, on the other end, have half the set of genetic information or half the set of chromosomes. They only have 1 of each pair. Think your sex cells, like sperm and egg. They have half of the genetic information, so they should have half the chromosomes. So like for us, our body cells are going to have 46 chromosomes, but our sex cells are only going to have 23. That should make sense again because your sex cells need to combine with another sex cell to make a full set of chromosomes or full diploid cell. You can see what that looks like here when we have 2 sex cells that have half the genetic information. The diploid cell they are going to create when they fertilize each other are going to have the complete set or diploid number of chromosomes. In this case, we have 2 chromosomes here, 2 chromosomes here. They're going to have 4 chromosomes in the somatic or diploid cell. You can see what that looks like here in this karyotype. You can see each pair of chromosomes comes from dad, one comes from mom. The purplish one comes from mother, the green one comes from dad. So you can see each set of chromosomes is going to have one from dad and one from mom. Again, this is a diploid cell since there are 2 for each pair of chromosomes. Now, something to remember when we talk about these cells are that 95% of a cell's life is going to be spent in interphase. And that should sort of make sense because we don't want your cells to be continuously dividing. Continuous division of cells is actually called cancer. And we're going to be talking about that next. So we want to really regulate how much your cells are going to go through mitosis. So most of the time, your cells are just going to be resting and going through their normal functions. They're not going to be dividing. If you think every one of your cells going through mitosis, you don't want to just keep growing and growing. You want to keep some parts growing, but not all parts. So only about 5% of your cells right now are going through mitosis. Now, there are proteins that regulate this. You can see that these proteins are going to make sure that these cells don't continuously go through mitosis. That we're going to do that at something called a checkpoint. But basically, they're going to check to make sure everything's functioning properly, that the cell isn't going to divide more than it should, and then it's going to keep on passing those cells through the cell cycle. If the cell is mutated, something's going wrong, the cell will, or the body will try to fix that cell, and if it can't, it will actually kill that cell. This helps regulate the cells from going into too much mitosis, like I said before, which can cause cancer. This is a cell end here. You can see that cell going through mitosis. This is what it actually looks like. I think it looks pretty cool here. The red part is the DNA. You can see the green parts are the spindle fibers. They're going to pull those chromosomes apart, but this is what mitosis actually looks like. Hello, everyone. We're going to start the next section with looking at a graph here. This graph is the total death or causes of death in, I think the United States this graph's for, but it's from last year in 2023. And as you can see, cancer is the number 2 leading cause of death in the United States. It's a terrible thing to get, but I like to preface this section with, yeah, this is a tough topic to talk about. A lot of students might have, something in their life where someone in their life where they're affected by cancer, and it can be really difficult. So I do want to kind of preface this with saying, yes, this is a difficult section to kind of learn about because it might affect you more than the other content that we go over. But I like to tell my students it's better to know about these things and be better educated so you can make better decisions if you're faced with the harsh reality of dealing with cancer. So what is cancer? Cancer is uncontrolled cell growth. This is just mitosis going uncontrolled. It is just mitosis going rampant, and these cells just continuously dividing. When we see cancer, we sometimes label it as a tumor. This is just a mass of cancer cells, but this is just what cancer is. It's just uncontrolled cell growth. Now, what causes cancer are called mutations. These mutations we're going to learn a lot more about later on in this year, but mutations are just changes in DNA. And these mutations can lead to cancer through 2 different ways. You can inherit these mutations through genetics, but you can also inherit these mutations or get these mutations through carcinogens, which we're going to talk about next. Now, it's not like you get one mutation and you automatically get cancer. It takes the cell a lot of mutations to develop cancer. You can see this normal cell going through mutations and cell damage. And then after a while, it's going to accumulate all of these mutations and it's going to become cancerous. So it's not just like 1 or 2 mutations. It has to be a lot of mutations for a cell to become cancerous. I also like to show this image where it's showing us the cigarette consumption in the United States back in the early 1900s and then lung cancer developing in men about 20 years later. This is showing that just because you do one thing one day doesn't mean that you're going to automatically get cancer, but you're increasing your likelihood if you keep on doing those things. So as the cigarette consumption increased, we saw it increase 20 years later because people just kept on smoking. They didn't realize, hey, this is causing an increase in likelihood that I'm going to develop cancer. When we realized this, we started better educating people and saying, hey, there is stuff going on here when you smoke. It's causing you to increase your likelihood of getting cancer. I also like to talk in this section about carcinogens. These are just cancer-causing substances that can lead to mutations. And as you can see in this chart, there's different levels of carcinogens. We label them usually 1 through 4. But like smoking is a high high molecule thing activity that can cause cancer. And there's other things that can cause cancer, but it's hard to classify them as a very likelihood of developing cancer. But just generally, we have different risks that we take when we expose ourselves to different molecules, compounds, or activities. Now, there's a lot of different things that can cause cancer that we know of. We talk about radiation from like X-rays. X-rays or UV radiation from tanning beds in the sun. This is why we put sunscreen on when you go outside. Chewing and smoking tobacco are a very high likelihood risk of developing cancer. Unhealthy drinking and eating can cause cancer. It again depends on the quantity and just the overall likelihood of what you're eating. But your environment also has a lot to do with it. Like if you're around car exhaust a lot, fumes, different work chemicals, these are all things that increase your likelihood of developing cancer. Now, it doesn't mean that if you just smoke one cigarette, you're going to develop cancer, but every cigarette you smoke or every drink or every UV radiation, whatever, every sunburn just increases your likelihood of then developing cancer. I like to end this chapter with about a section about the effects of cancer, like why is cancer deadly? So what will happen is that what the cancer will actually do is it will shut down the organ where it's located. You would think like, oh, it's just more cells. It's just, doing whatever those cells normally do. But those cells that are cancerous actually won't function for the cell or the organ that it's in. So if it's like a liver cell, it's going to shut down the liver because those cells are going to take away nutrients and energy from the liver and will actually shut down the liver. And if you don't have a liver, you have a high likelihood of passing away. So what actually happens is this cancer will actually shut down or stop the function of the organ that it's in. Whenever cells that are cancerous travel to other locations, this is called metastasis. And this is what doctors and just anyone who deals with cancer try to prevent, is because once you start seeing that cancer start to spread, it's much more difficult to fight. So if you have lung cancer and it develops and it spreads to the liver, it's much harder to fight two types of cancer than just one type of cancer. This is what this is what the doctors try to prevent, is the metastasis of the cancer cells. Now, when the cell does become cancerous, what your body can do is it can send it through something called apoptosis. This is just controlled cell death, where the cell will actually just die. And it will try to your body will try to kill that cell in order to prevent it from becoming cancerous. Now, this doesn't always work because, again, people develop cancer, but this is one way our bodies have developed or evolved a way to stop cancer. This is a cell going through. I'm going to show this just quickly. This is a cell going through apoptosis. You can see it just kind of breaking down, and then eventually it's going to almost kind of like pop. The membrane will actually start to fission here and actually split up, and that cell will go through apoptosis. Here it is. It's going to kind of just go away. And this is, again, a way that the cell or the body is going to stop a cell from becoming cancerous as it starts to spread. It's a pretty interesting process. This was, I think I found this on social media. Somebody records these cells and their processes. There we go. That's what cell kind of death kind of looks like. We're going to move on. There we go. So what do we do about cancer? So we treat it with something called chemotherapy. This is a kind of a harmful way to kill cells. And this is kind of a thing of the past. Is we're starting to move or away from this. This is where we're just killing a lot of cells. We're trying to kill the cancer cells, but we also kill the healthy cells. And this is just what we've had to do in the past because we just didn't have a better way. This is very invasive, what's called, just very harmful to the person. We use sometimes radiation, sometimes we even use surgery to remove these cancer cells. What we've been doing in the more recent years is something called immunotherapy, where we're trying to train our immune systems to target and fight those cancer cells. And this is a much better way of fighting this cancer, but it's a lot more costly as well. This is just more precise and more targeted to a person rather than just, hey, we're going to use this for everyone. So this is something in the future that hopefully if somebody that you know or yourself develops cancer, that you'll be able to take part in, because this is a much better, healthier, and just effective way of targeting and killing cancer. I like to end with this this graph because I feel like I talk about cancer and students might get fearful, but realize that yes, cancer deaths have been going up, but it's not that they've been actually increasing. So what we're doing a better job at is we're doing a better job at identifying when somebody has cancer. Back 200 years ago, we really didn't know what cancer was, and a lot of times when somebody died, we didn't know what they died of. So we're just getting better at identifying when somebody has cancer. And if you look at this blue line, you can see it's the cancer death rate per 100,000 people has drastically decreased. Where 100 years ago, cancer or getting cancer was almost like a death sentence, now we have much better ways of fighting off this cancer, and people are surviving with cancer more and more every single year. Because again, we're identifying it earlier, we're finding better ways to fight it. So don't think that cancer is increasing. We're just getting better at identifying it and also fighting the cancer.